So this is a tutorial session on empirical Bayes. So empirical Bayes methods are often, there's kind of a history of motivating them via baseball examples. So I don't know pretty much anything about baseball, so you don't need to know anything about baseball in order to appreciate this example. But this example comes from a blog post by David Robinson, the data do anyway. And it's a data set on what's called batting averages from some time ago, 1900. And so what's being shown here is a histogram of the batting averages of all the players who played that season. And okay, so a batting average is basically in baseball, you go and stand in front of someone who throws a ball at you, you try and hit it, and roughly speaking, the proportion of times that you successfully hit it is called your batting average. So if you hit it 20% of the time, your batting average is 20%. It's a bit more complicated than that, but that's the idea. And so what you can see here is there's a range of batting averages, and some players have batting averages in these data that are below even 10%, very close to zero, and others are even exceeding 40%. And anyone who does know something about baseball will tell you that these are crazy batting averages, the biggest ones and the smallest ones here. No one actually gets a batting average of over 40%, and no one in these leagues are going to get a batting average under 10% or even under maybe 15%. So what's going on? And the answer to that is that these are, when you look at the data, what's happened here is that some players just didn't play very often. So maybe they only played twice, or they only went even at bat twice, and they missed both times. And so then their batting average is 0 divided by 2, which is 0. Or someone may have, if they went up twice and they hit once, they would have a batting average of 50%. Right? So essentially what's going on here is that these are, quote, estimates of the batting average for each player. They're the estimates of what would happen in the long run if this player played many times. But they're point estimates, and in some cases they're very inaccurate. Okay, so the idea behind shrinkage is that we should use, well, the idea behind empirical Bayes shrinkage anyway, is that we should use what we know about batting averages to improve our estimates, especially these very noisy estimates. Right? So what I've just, so the idea is that essentially if we know that batting averages are typically between 20 and 30%, we should be moving the ones that are very big, we should actually estimate lower, and the ones that are very small, we should actually estimate higher. We should say that these guys probably just got unlucky, and if they were to go batting again in next season, let's say, and we had to estimate what we thought they would achieve, it would be higher than 0. And vice versa, the ones that are at 40%, if we had to estimate what they'd achieve next season, we'd guess less than 40%. So that's the basic idea of shrinking. Is the name comes from moving things, the extremes get shrunken towards a center. So how much should we shrink? Well, essentially, the answer I want to give you to that question is that it depends on what we know about batting averages. So the red line here, the line that just came up, it is red on my screen, is showing a hypothetical kind of distribution of actual batting averages. So imagine that we had some information on what typical batting averages looks like. What's the distribution of batting averages? We could use that information, and we could combine it with the information we have to decide how much to shrink these estimates so that they look more like that overall distribution. Okay, so that's just the intuition behind the mechanics. What's the idea? What's the essence of this problem? The essence is that we've measured lots of similar things, that is, a whole bunch of batting averages. They're similar to one another in that sense. They're the same thing, but they're for different people, and they're measured with error. And the idea is that we want to use the overall distribution of these things to improve the estimate of each individual one. And in outline, the empirical Bayes approach will be a two-step approach. The first will be that we'll use the data we've got to estimate what is the overall distribution of these, in this case, batting averages. That is, to estimate that red line that we might not otherwise know. And then once we've estimated that, we're going to combine that information with each individual measurement to improve the estimate of the batting average for each person. So that's the essence. So this, I'm now going to try and encapsulate this essence in a simple model that's going to be based on normally distributed data. Now actually, there's a slight wrinkle here, which is the baseball data aren't actually strictly normal. But this is a simple model, and so I'm going to assume normal errors for simplicity. So this is a model for the idea here. We've got observations x, or xj for each j, and they're noisy observations of some underlying thing, theta, j, and they've got some known standard error, Sj. Okay? So the Sj represents essentially the precision of the, or captures the precision or the error in the estimate or the measurement. X is the measurement, and theta is the thing we don't know. And the next layer is to assume that the thetas come from some distribution. So the thing of the thetas is the true batting averages. They come from some distribution. I'm going to call it G. That was the red line in my picture. And so mathematically, the empirical Bayes approach proceeds in two steps now. The first is that we need to estimate G, that's that distribution of all batting averages. And then, secondly, having estimated G, we're going to estimate theta j, taking account of both that underlying distribution and the observation xj that's specific to unit j. Yeah? So it's like combining background information, G, with specific information on that person, xj. So if you want a picture, you can think of it like this as some distribution of effects or batting averages at the top, G, and then there are thetas, which we don't know, but we're interested in, and they're independent in this model draws from G, and then the thetas are yielding observations X that are noisy observations of the thetas, right? So what we see is the X's at the bottom, and what we're interested in is the thetas. And the idea is we're going to do this in two steps. Firstly, we're going to use the x's to estimate G, taking account of the fact that we don't know what the thetas are. And then once we've estimated G, we're going to assume it's right. And that's the empirical Bayes approach, part of the approach. Assume your estimate for G is right. You've got your x's at the bottom, the G at the top, and in between the thetas are going to get estimated. So each theta now, theta 1 gets estimated taking account of G and x1. Theta 2 gets estimated taking account of G and x2, etc. So that's a visual. Okay, so I've really tried to emphasize the kind of conceptual idea, but in order to actually implement this in practice, we're going to need specific methods to estimate G and estimate theta, taking account of G and xj. So in brief, the way we're going to do this is to estimate G by maximum likelihood under this model. That is, by choosing the G that maximizes the probability of the observed data, X. And we're going to estimate theta by computing its posterior distribution given using G as the prior distribution and P of theta given X. Sorry, that should say P of X given theta. So Px given theta, that's a typo. The likelihood, combining the prior and the likelihood. Essentially, this is straightforward Bayesian inference for theta once we've estimated G. Okay, so I'm going to talk a little bit more about those two steps. Maybe, I've got a curly G there. Little g is a member of this family. Curly G, what does curly G represent? Curly G represents some family of, quotes, prior distributions. Maybe I should say G is sometimes referred to or thought of as the, quotes, prior distribution, because it's when we do the second step, that's the role it's playing in the inference for theta. So the family of prior distributions, curly G, will obviously influence how hard or easy this step 1 is, and actually also how hard or easy step 2 is. So here's one simple but flexible family that turns out to be, I think, quite useful, but there are many others, but this is just one example of what you can do. So this is what's known as the scale mixture of normals family. So the scale mixture of normals is a mixture of normals where each component of the mixture has a different variance. So in this picture here, in the formula here, you can see it's a mixture of capital K normals. They have variances sigma 1 squared, sigma 2 squared, up to sigma k squared. And you can think of those sigma squareds as varying from very small to very big. And capital K as being a large. So what we're going to do here is take a mixture of a large number of components. Some of those components have very small variance. Some of them have very large variance. And how we're going to estimate G is by estimating these mixture proportions, pi. And the reason to do this way is it's a flexible family that leads to a convex optimization problem. So estimating pi is a convex optimization problem. So it's basically an easy optimization problem. Let me give you a picture of what these distributions, when I say they're flexible, here's a mixture of 3 normals. They're all centered at 0 here. One's got a small variance, one's got a medium variance, one's got a larger variance. And when I mix them together, I get this funny looking distribution that has lots of mass near 0, but also a long tail of mass away from 0. So you can imagine, that's using only 3. You can imagine even just using the 3 distributions, you could get different behavior depending on which one you put heavy weight on. But by using a larger number of components, say 20 different components, you can get a very flexible family of distributions. So just to kind of recap then, now estimating G, if we are prepared to assume this family, it becomes a convex optimization problem, and there are simple algorithms for doing it. Actually, there are lots of other families you might use, and even though they don't lead to convex optimization problems, they lead to optimization problems that are often not that hard. So another family you might use is a mixture of 0 and a normal. That's sometimes called a spike and slab prior family. So a mixture of zeros and non-zeros. It's kind of motivated by the idea that in many applications you might think that a lot of the effects, quotes, that you're looking at are 0. Of course, in the baseball example, that's not true. I should say in the baseball example, you wouldn't center everything on 0. Right? But it turns out that in genomic applications, you often think of the effects being centered at 0, which is why I'm focusing on that case. So estimating G comes down to estimating these mixture proportions in this case, which is easy. And then combining G with the likelihood is a straightforward application of Bayes' theorem. I just want to visually show you what's going on here. Sorry if my text seems to have been cut off at the bottom. But so visually what's going on here is that the black is meant to represent a prior distribution that we've just estimated. That was our G. And the red, the one on the right, represents the likelihood. So in this case, maybe we've had an observation of xj of about 1.5, and it's got a standard deviation of somewhere, I don't know, a half or something. All right, so that's that likelihood you've got there, the red line. And when you combine that prior and the likelihood using Bayes' theorem, you get the posterior distribution, which is the green line. And so the idea, if you're not familiar with Bayes' theorem, is it multiplies together the prior and the likelihood, and the result is that the posterior distribution is appreciable only where both the prior and the likelihood have some appreciable mass, which in this case is, in this case, in fact, the prior and the likelihood kind of are a little bit contradictory to one another, because one's big where the other's small and the other's big where the other's small. So what you get when you multiply them together isn't quite obvious, but I can show you it's the green line. So what you can see is that the mean of the green distribution is moved towards 0 compared with the red. So the red was the likelihood. The peak of the red was the maximum likelihood estimate. That's like kind of the point estimate of the batting average, if you like. And the result of Bayes' theorem is you've shrunk the distribution towards the prior, which in this case was the black line, centered at 0. So how much you end up shrinking now, these estimates, depends on both the prior distribution and how kind of spiky it is. The more spiky it is, the more you're going to weight your prior, and the likelihood and how spiky it is, or how big the standard errors are. The smaller the standard errors, the more you're going to weight your likelihood. So in the baseball example, players who've been at bat a lot of times, you've got more information about them, and so you weight their data more than the prior. Players who've only been at bat 3 times, you have very little information about them, and so you end up weighting the prior rather than the likelihood. So that's illustrated by this case where the red line now is much flatter. That's meant to represent a data point which has a large standard error. You don't have as much information about it. And when you combine that red line with the black line, you get the green line, which is basically on top of the black line. Yeah, so in this case, although the red line has a peak at the same point as the previous red line, the green peak gets shrunk much, much more strongly towards the prior, which in this case is centered at 0. So the amount of shrinkage that you get is determined by both, or depends on both, the prior G, which is estimated from the data, and it also depends on the standard errors. Observations that have larger standard error get shrunk more because you weight the prior more heavily. And this all happens automatically. You don't have to. I say you weight the prior more heavily. Bayes' theorem Bayes' theorem multiplies the prior and the likelihood together, and if the likelihood's flat, then you just get the prior back. That's kind of the intuition. So this is a very flexible framework for thinking about the way of combining information from multiple measurements and improving estimates of any given one of them. You can implement this kind of idea for a wide range of prior families. G, I mentioned the spike and slab point normal prior, the scale mixtures of normal prior. You can do many other possibilities, including ones that do not necessarily have a mean at 0, or could be non-negative in certain applications if the things that you're looking at are non-negative. And maybe I'll just comment, I focused on in the baseball example, I focused on the idea of shrinking the estimates, but you actually get not only point estimates that take account of the prior and the data, but you also get interval estimates, or actually whole distributions on these, on what's the plausible values for theta. Okay, so despite this, it's simple, it's flexible, it has lots of potential applications. I would say maybe in practice, it's these ideas are not as widely used as they could be. So my encouragement to you is to think about cases or be on the lookout for situations where you might want to use some kind of shrinkage type method to combine information in this way. And anyone who's seen Meet the Parents may appreciate this reference. I'm going to try and tell you that you can shrink anything with standard errors. So if you see a bunch of things and they've got standard errors, you should be thinking about maybe you could shrink them. Shrinking one thing with a standard error isn't so recommended maybe, but if you've got more than one, you might be in business. Let me give you an example here. So this came from a data set we were looking at. It's called the GTEx data set. Comes from the GTEx project. Comes from genes, tissues, and expression. So this project collected gene expression of lots of genes in lots of different tissues. And what we were interested in is that some genes seem to show correlations in their expression between different tissues. In other words, how expressed that gene was in your heart was also, it seems, correlated with how expressed it was in another tissue like your liver. Whereas other genes showed much less correlation. So what's shown here is just a point estimate for every pair of tissues, and you don't need to see which tissues are which for now. I will mention that the ones at the top left that show the patch of red are all different parts of the brain. Okay. So, but what this heat map's showing is for every pair of tissues, what's the correlation of the gene expression for this specific gene, VSIR, in that pair of tissues. And you can see that many of the correlations are strong and positive, so particularly pairs of brain tissues often seem to have strong positive correlations. But there are other, the blue dots, which show pairs of tissues that appear to have negative correlation in their gene expression, even pretty sizably negative, close to negative one gene correlation in their gene expression. And that seems a bit weird, right? So that would seem like this is saying that if you're highly expressed in this tissue, then the same gene tends to be lowly expressed in another tissue. Seems a little unexpected. And when you go and look at what's going on here, almost all the blue, or certainly the very dark blue dots, are due to the fact that some pairs of tissues do not have many samples in common. So what they've got is they've got about 1,000 individuals here and 50 tissues, but no one person has been measured at all 50 tissues, right? So some tissues have only 10 samples, and others might have hundreds, and then for every pair, we only have the tissues that were measured in the same individuals, which might be as small as 0 in some cases, right? Actually, I don't think there are any zeros here. I got rid of that, but some of these sample sizes are very small. So we're kind of seeing something similar to the baseball example, right? Where when people were only at bat a few times, their batting averages were very unreliable. When you've only got 6 samples here and you're estimating a correlation between them, those estimates are very unreliable. And so we can kind of clean these estimates up by applying empirical Bayes shrinkage. Now, these are correlations, and correlations are between -1 and 1, so they're not strictly normally distributed, but a quick, just to briefly say, you can get around that problem by a transformation known as the Fisher transform, that essentially it's this half log 1 plus r over 1 minus r, where r is the correlation. So that transforms an observed correlation into something that's approximately normally distributed with a standard error that's given here. It depends on the sample sizes, you can see. So the standard error, Sij, looks roughly like the square root of 1 over the number of samples. And and this is what you get when you just apply the vanilla empirical Bayes normal means idea to shrink these correlations at this gene. And what you can see is that it's preserved the very dark red in the brain, which is good because we probably believe that's real. That seems biologically plausible that the expression of one part of a gene in one part of the brain might be correlated with its expression in other parts of the brain, but it's largely removed all those blue ones. Yeah?
So what you gain by this transformation is only that you can now apply bounds on that, I mean, to like p-values to really...
Yeah, what you gain from the transformation is just the ability to apply our normal means model, which assumes normal errors. So that's what you gain.
Yeah.
Yeah. So the transformation is really an additional step here to allow us to apply a model that assumes normal errors. An alternative would be to devise a version of the empirical Bayes normal means model specifically for correlation data, right? You could try and, the framework's not specific to normals, but we've got a simple implementation for the normal case. And so if you can transform your data to fit the normal assumptions, then that's a simpler way to go than to build a whole new method. This is just to show another gene. You get, not all the genes are the same. So in this gene, there are very few strong red signals. So what happens is that most of the estimates go to 0 here. So what's happening here is that in this example, the before picture here shows some very strong dark red in tissues that had relatively large sample sizes, so we kind of believe them, right? So when we learn the G, the prior distribution for this example, it has some mass on large correlations near 1. Yeah. And so when we shrink them, we combine that G we learned with the data, it looks at the correlations near 1 and it says, oh yeah, that's pretty plausible because actually my G that I estimated has mass near 1 and it leaves those alone. Whereas in this example, what happens is that there are even though there are a few dark red points, they happen to cut, they lie at pairs of tissues that have very small sample sizes, and so we don't really believe them. And so when you estimate the G for this gene, it looks different from the G for the previous gene. It has most of its mass near 0, and so then the result is you get much stronger shrinkage towards 0. It's different behavior. Yeah.
On the previous image, like you have a vertical kind of dark red line in the before, right?
Yeah.
And that's gone. Is that, just in that context, is it a good thing that you got rid of that? Do you think that's also like a false signal on the red side? Like in the before, the top left corner is preserved, which is a good thing.
Yes.
And a little bit to the right of that, there's kind of a long red block.
Yes.
And it eliminates that, which like, do you think that's a good elimination? Like
yeah. So we'd have to go back and check the sample sizes, but my guess is that that's a tissue that has relatively small sample sizes,
as well,
which is why it got removed. Because the method doesn't know that these are all brain and this one's not brain. It doesn't know that. It's not using that information. Arguably, one should use that information, but that would, we're not building that information in. So the method's kind of unbiased in that sense. So the fact that it's removed that one and left the brain is not because it knows that those are brains, and it must be based on sample size. Yeah. And this is a extreme example the other way where there's a lot of red, including in things that have very large sample size. So the estimated G, it learns that this gene actually shows many strong correlations. Because every gene is different, essentially. Some genes do show strong correlations across tissues, and others do not. And so you get different behavior depending on the tissue. Okay, another example of a potential application of these ideas is multiple testing and false discovery rate. So, I would characterize the canonical genomics experiment as actually measuring lots of things with error. We do that a lot in genomics. Oftentimes you end up calling the estimated effects beta hat. They're an estimate of some underlying beta. They've got a standard error. And our goal in many cases is to identify which beta are significantly different from 0. So one example here might be gene expression studies where you're looking at the effect, well, maybe I'll say in later we're going to be looking at gene expression studies that are looking at the effects of genetic variants on gene expression. So the beta J will be what's the difference in gene expression between one genotype and another genotype. So beta J is like an effect of genotype on gene expression. And we might think that most genotypes have no or very small effect on gene expression, so most of the beta Js are 0 or close to 0, and we want to identify the beta Js that we can confidently conclude are different from 0. So this is sometimes referred to as the multiple testing setting, where you're testing lots and lots of things. And this situation is kind of ideally set up for the EB&M, empirical Bayes normal means problem. You've got lots of measurements. The beta hats here are our x's. The standard errors are, of course, the standard errors. And the thetas are just the betas that we're trying to estimate here. And you can use the ideas from EB&M to instead of focusing on point estimates here, we might be interested in questions related to whether beta is 0 or not. So, for example, the local false discovery rate is looking at the posterior probability that beta is non-zero, or what I call the local false sign rate, which has a slightly more complicated formula here, but is essentially getting at the question of can we be confident in the sign of the effect, which is a little bit more robust, it turns out, than asking whether an effect is 0 or not. You can get more robust answers if you ask a slightly different question, which is, let's just assume everything's non-zero, and ask, can we be confident in whether it's positive or negative? I'm not going to dwell too long on this example, but just if you're familiar with multiple testing and maybe familiar with the Benjamini-Hochberg, which is a widely used procedure in multiple testing, I just want to kind of compare and contrast the empirical Bayes approach with the Benjamini-Hochberg-based approach. So both approaches are what I might call generic. So Benjamini-Hochberg, if you're not familiar with it, essentially takes a whole bunch of p-values from a large number of tests, and it uses using those p-values in a clever rule, it spits out a list of which ones you can consider significant at a given false discovery rate. So the nice thing about this is you can apply this wherever you've just got a bunch of p-values. So this idea is the same, except instead of p-values, you need a beta hat and a standard error. Okay, so you need two numbers instead of one, but oftentimes where you've got p-values, you've got them from a beta hat and a standard error. So oftentimes this will also be easily applicable. So it's generic in the sense you can apply it whenever you have a bunch of beta hats and corresponding standard errors. So that's nice. Unlike the Benjamini-Hochberg, the empirical Bayes normal means approach explicitly models this G. And so this has some advantages. Particular advantages, it can account for differences in the standard errors. That is, if some measurements have much less precision than others, it can account for that because it's using the standard errors. And it can also provide estimates of the effect size, whereas the Benjamini-Hochberg is just significant, not significant. So, and I have a paper on this if you're interested in more details. Maybe I want to say, though, it's my last example, is that although I think there are some advantages to the empirical Bayes approach over the Benjamini-Hochberg approach in this simple multiple testing setting, in practice I think the gains probably are more important in a slightly more complex setting, which I'm going to tell you about now as the last part of my talk. So the extension is to multiple testing where you've got multiple treatments or multiple conditions. So in genomics, many times when you're doing lots of tests, you're maybe testing lots of things. You're not only testing them in one condition, but in multiple conditions. So for example, in genome-wide association studies, you might test for genetic variants associated with, I don't know, a trait like height, but you also might test them for association with other related traits like lengths of different bones, weight, various anthropomorphic traits. Right? So you often are looking at multiple traits. In the GTEx study that I mentioned earlier, we were particularly interested in the effects of genetic variants in different tissues. So we were interested in testing each genetic variant for whether it had an effect on gene expression of a given gene, but we were interested in doing that in 50 different tissues, 49 tissues. Right? So when we're doing this analysis, we would like to, just like we wanted to kind of use the overall distribution of baseball batting averages to improve our estimate of any one, here we would like to use the distribution of effect sizes, and in particular any sharing of effects that might occur across conditions or between conditions to improve our estimates and inferences for each tissue. So this might be particularly relevant for a tissue where you have less data, for example. It's harder to sample, so you've got fewer samples. But if you can learn that that tissue tends to behave similarly to another tissue, not exactly the same, but similarly to another tissue where you've got more samples, you could kind of leverage that to improve your inference for the smaller sample tissue. Yeah, I don't think I need. OK, so this is a picture of the z-scores we get from an analysis of eQTLs in the GTEx study. So the rows here are SNP gene pairs, genetic variant gene pairs, and the columns are tissues. And if you can't see the plot very well, that's fine, because the point is that most of it's close to 0. Most of the z-scores are, quotes, not significant. Right? But I've ordered them in a way that the more significant ones are at the bottom. And you can see just visually that there seems to be some sharing of the significant effects, right? That maybe oftentimes when there's significant in one tissue, it often is significant in many tissues. And there seems to be a bunch of tissues, which happens to be the brain tissues again, they're in a slightly different position in this plot, that you might be able to see that seem to maybe share more effects than, and be slightly different to the other tissues, behaving slightly different. So the intuition is that you can look at these z-scores, and maybe you could learn that kind of fact from these z-scores. That's like learning the G. You can learn that, oh, it seems like the brain tissues are sharing effects with one another more, and maybe other tissues are also sharing effects, and trying to learn those patterns from the z-scores. That's the intuition here. So the picture is exactly the same as before. You've got a distribution G, which is capturing the distribution of the true effect. You've got the true effect theta that you don't know, and you've got x's, which in this case are the z-scores that are observed noisy observations of the true effects. But now, unlike the normal means problem I mentioned earlier, now the thetas are vectors. So each theta here is a vector of length, the number of tissues. I think it's 44, yeah, 44. So G is now a prior distribution on things of length, on vectors of length 44. So so it's definitely harder to estimate G now, and whereas in the empirical Bayes normal means problem, I said there's lots of different prior families you might use, blah, blah. In this case, there's only one prior family that we've actually implemented, and it's definitely not as flexible as the kind of fairly non-parametric prior families we used in the univariate case. But anyway, this is what we essentially use a mixture of multivariate normals here, which is what I've got at the bottom. So in the univariate case, what I showed you before was a scale mixture of univariate normals. So we're kind of extending that idea using mixtures of multivariate normals to model our Gs. And now the hardest part of this is that now we need these covariance matrices. The UK here are now covariance matrices that are capturing the sharing. So I'm not going to talk about how you fit these models, but I'm going to show you some results of what kinds of things it produces when you apply it to data. So this is one of the covariance matrices that comes out of our estimation pipeline. And so what you can see here is I hope you can see that there's two groups of tissues with very strong covariance. The first group is brain-related tissues, that's the one at the top, along with testes and pituitary, which happen to be correlated with brain in terms of the effect sizes. And then there's all the other tissues that are kind of forming another clump. And what might be less obvious, but is true, is that actually even in the outside the block there, those correlations are also quite positive. So even the brain and the non-brain tissues are also, the effect sizes are quite correlated. They're just not as correlated as within the non-brain tissues or within the brain tissues. This is just one of the covariance matrices. We're using a mixture. So what the mixture can do is pick up other patterns that are maybe, this is kind of the most common pattern in the data, but there are other patterns that exist. So for example, there are some effects that seem to occur only in blood and not in the other tissues. So there's a covariance matrix I'm not showing you that captures effects that essentially only occur in blood. And this is what happens when after you've estimated your G and you apply it to your data, combine the prior with the data. So what's shown here on the left-hand side is the beta hats and the standard errors for each tissue. So it's beta hat plus or minus 2 standard errors. So each row is a tissue. The point estimate beta hat is shown as a blob, and the gray line shows plus or minus 2 standard errors. So what you can see is that this is for one row of that z-score matrix. So it's for one genetic variant testing for association with one gene, but in all 44 tissues. So what you can see is that this genetic variant seems to be reasonably consistently associated with gene expression in the brain tissues. The brain tissues are like the third, fourth, fifth, sixth, about 10 tissues there. Right? The ones that are shifted to the right. And in most other tissues, there's not a significant effect, but if you squint, you can maybe convince yourself that the dots tend to be on the right of 0. So maybe there's a hint that there's some effect in most tissues, but it's small in the non-brain tissues. And what you can see is that the posterior really cleans this picture up. The posterior standard deviations, so these are now point estimates plus or minus posterior standard deviations. And the posterior standard deviations are much narrower because of this effect of combining information. When you combine information, you get more confidence, more certainty. And you can also see that most of the tissues now have positive effects. The effect is kind of borderline, quote, significant in most tissues because of this fact that even though most non-brain tissues were not independently nominally significant, there's this kind of overall trend towards a positive effect. I will, in the interest of time, I will skip over to the final couple of slides. This just says, nice thing that comes out of this is that indeed similar tissues have similar effect sizes. So actually this is maybe going back to one of the advantages of the empirical Bayes approach over, say, just Benyamini-Hochberg, is that you get effect size estimates. So this is kind of trying to lean into that and saying, oh look, actually the high level summary is there's a lot of sharing of effects across tissues, but if you look at the effect sizes, you see that more similar tissues tend to have more similar effect sizes. So if you just ask about sharing, you don't get that kind of insight because everything's kind of shared, or a lot of things are shared, I should say. Lots of things are shared. But if you look at effect sizes, you really start to see, oh yeah, the two different skin tissues have very similar effect sizes. Okay, so in summary, empirical Bayes approaches combine information across many measurements to improve estimates and measures of uncertainty, and you can shrink anything with standard errors. And if you want some more of those various papers on my website, I'd be happy to answer questions.